For the sake of simplicity, the plasma membrane shown in this video will exclude proteins cholesterol, glycolipids, and glycoproteins. Please note that they are present nonetheless. Plasma membranes are selectively permeable, meaning some things can enter and exit the cell. But some are harmful substances that need to stay out, while organelles and structures need to stay inside of the cell. Cells need to receive nutrients and oxygen constantly. Likewise, they need to get rid of CO2 and waste. Fortunately, there is a process that happens naturally. It's called passive transport, and it doesn't require any energy. Passive transport works by using concentration gradients, which is the difference in concentration in a given area causing molecules to move from an area of higher concentration to an area of lower concentration. Notice how oxygen, represented by the red spheres, is found more outside of the cell in the beginning, and likewise, carbon dioxide is found more inside. Thanks to passive transport and the use of concentration gradients, the molecules move to achieve equilibrium. This occurs when molecules are found in equal concentration across an area. Please keep in mind that living organisms are in constant flux. As those oxygen molecules enter the cell, more will congregate outside. Likewise, more carbon dioxide will build up on the inside of the cell as they get ready to diffuse outwards. The permeability of a structure is its tendency to allow a given substance to pass across it. Phospholipid bilayers have selective permeability. Small, uncharged, or nonpolar molecules move across phospholipid bilayers quickly. Charged, larger, or polar substances cross slowly, if at all. In fact, small ions, like sodium ions, can't pass through unless through a special channel protein. That's because the positive charge can't react to the uncharged phospholipid tails, causing them to repel. Now, this is, in my opinion, one of the most important concepts you should learn in biology because it helps explain not only how stuff moves across cells, but how urine is formed and how blood delivers nutrients into and out of our bodies. So how does passive transport work? It works by this physical phenomenon known as diffusion. And diffusion is the movement of molecules across an area of higher concentration to an area of lower concentration to achieve equilibrium of concentration. Before we continue in more detail about diffusion, we need to define the components involved. First are solvents. Solvents dissolve solutes. So a solvent, in this case, would be water. Water has the ability to dissolve substances and get them to mix together. Those substances that dissolve in solvents are called solutes. Think of it as salt. Solutions are a mix of solvents and solutes. So salt water would be an example of a simple solution. Materials can move across the cell membrane in different ways. Passive transport does not require any input of energy. Active transport requires energy to move substances across the membrane. We'll discuss those later. Diffusion is a type of passive transport, so let's demonstrate how diffusion works using a simple setup of food coloring and water. In this demonstration, water is our solvent and we're using highly concentrated food coloring. Notice in the beginning that the food coloring heads towards the bottom of the jar. And as we speed this up a bit, the concentration of the red food coloring increases at the bottom, decreases at the top, and eventually will start to go towards the top to try to achieve equilibrium. Eventually, it looks like this. No energy was needed for this entire reaction to occur, and now the entire solution has achieved equilibrium of concentration. Diffusion requires concentration gradients which are formed between two different areas. These provide pathways for molecules to travel. A difference in solute concentration creates a concentration gradient. Molecules and ions move randomly when a concentration gradient exists, but there is a net movement from high concentration regions to low concentration regions. Equilibrium is established once the molecules or ions are randomly distributed through a solution. Molecules are still moving randomly, but there is no more net movement. Try to imagine these molecules going in and out continuously. It's just that I'm limited in displaying all of that due to the technology I'm currently using. There are many factors that affect diffusion, which I'll discuss next, but one example is size. Larger molecules, regardless of the concentration gradient that exists, still cannot pass through easily through the plasma membrane, simply because they just can't pass through the physical barrier. So here are some factors that affect the rate of diffusion. Temperature. Warmer temperatures tend to increase the rate of diffusion, while colder temperatures decrease it. Remember that colder temperatures cause membranes to shrivel, making it harder to move substances across a smaller surface area. And brings me to the next point, the surface area of the plasma membrane. When you have smaller surface areas, there is less rate of diffusion, whereas a larger surface area provides more opportunities for diffusion. For example, inside of our lungs, we have the cells take up a large amount of surface area to help increase the amount of gas exchange that occurs. If there is a greater difference in concentration gradient, there will be a higher rate of diffusion. The closer the concentration gradient gets to equilibrium, the slower the level of diffusion becomes. As I mentioned earlier, the size of a molecule, if it's heavier or larger, it's going to take longer to diffuse, but if it's smaller or lighter, it's going to diffuse much quicker. Higher solvent densities reduce diffusion. Notice how doctors always say rest and drink plenty of fluids? These fluids increase the rate of diffusion so cells can get what they need to fight off disease and repair themselves. Also, when dehydrated, cell function slows down. As mentioned earlier, nonpolar, lipid-soluble materials will diffuse better than polar, water-soluble materials because of the molecular structure of the membranes. The thicker the membrane, the slower the rate of diffusion. And I mentioned earlier about the air sacs in the lungs. Not only do they have a larger surface area, but they're also, the tissue that makes that up is very thin, which allows for more gas exchange to take place. Finally, the rate of diffusion decreases over greater distances. So now I want to focus on facilitated transport. So what do you do with larger, charged, or more polar molecules? Remember the proteins from earlier? Some of them serve as either channel or carrier proteins, which help to transport those sorts of molecules across cell membranes. This works the same as simple diffusion, as the concentration gradient still goes from high to low and it requires no ATP on the part of the cell. Channel proteins are selective of the molecules that are appropriate size and charge. One special type of channel protein is called an aquaporin, which helps to move water across the plasma membrane. In this case, I'm using glucose. Now notice again, the end goal is to achieve equilibrium. Carrier proteins are membrane proteins that are physically bond to specific substances. They change the shape and move them across. What you just saw was the wrong molecule coming in contact with the carrier protein, and so no reaction occurs. But when the appropriate one bonds to the carrier protein, it brings the molecule in. Some carrier proteins act as pumps, which require ATP, and is a form of active transport, which we will discuss later. So osmosis is the diffusion of water across a selectively permeable membrane. Technically speaking, diffusion occurs with or without membranes. Like diffusion, osmosis is a form of passive transport and requires no ATP on the part of the cell to operate. Water moves quickly across lipid bilayers. The movement of water is a special case of diffusion called osmosis. Water moves from regions of low solute concentration to regions of high solute concentration. In other words, water always follows the greater solute concentration, depending on which side of the membrane we're looking at. This movement dilutes the higher concentration, thus equalizing the concentration on both sides of the bilayer. In this example, I've added electrolytes to represent solutes. Electrolytes help with water balance anyway and represent many ions we get from our diet, such as potassium, sodium, and magnesium. Water can't pass through without the aid of aquaporin proteins, pictured in orange, because of water's polarity and incompatibility to pass through nonpolar tails of phospholipids. Since there are more solutes inside of the cell than on the outside of the cell, water will enter the cell at a higher rate than it leaves. And try to remember that rule.
Water always follows the greater solute concentration.
And the whole point is to try to achieve equilibrium of concentration on both sides. Osmosis can only occur across a selectively permeable membrane. So while solutes can't pass through membranes, sometimes water can, as indicated by these two beakers. However, the concentration of a solution outside of a cell may differ from the concentration inside the cell. The tonicity, or strength of a solution, can have big effects on the cells themselves. So let's talk about that. Tonicity describes how an extracellular solution can change a cell's volume by affecting osmosis. Osmolarity describes the solution's total solute. If you remember the rule that
water always follows the greater solute concentration,
you can predict how water moves in and out of cells. There are three general types of osmolarity that cells are exposed to. Let's assume each cell cytoplasm is 99.1% water and 0.09% solutes. And solutes can include anything, electrolytes, protein, waste, things that dissolve within the cytoplasm of a cell. In isotonic solutions, a cell's extracellular fluid solute concentration matches that of the cytosol within the cell. This causes no net change in the volume of the cell as water enters and exits the cell at the same rate. In hypertonic conditions, cells are exposed to extracellular fluid that has a higher solute concentration than the cytosol. Since equilibrium must occur, the cell loses water to the surrounding environment and shrivels as a result. This can lead to dehydration and loss of diffusion of solutes across membranes. Finally, hypotonic solutions occur when cells are exposed to an extracellular fluid that has a lower solute concentration than the cytosol. Since equilibrium must occur, the cell gains more water from the surrounding environment and swells. If too much occurs, the cell membrane can burst. I want to show you all a really cool thing you can try at home with eggs. Eggs and osmosis. Eggs are essentially a really large cell, so they make the perfect model for this. What I'll be doing is I'll be taking these 3 eggs and I'm going to have to insert some vinegar. Now, the reason why we do this is the eggs have a calcium carbonate shell. And in order to make sure that that shell is dissolved, we're going to put vinegar in there to help do that. And so here's the chemical formula. It's vinegar plus calcium carbonate shells will create carbon dioxide gas. And we'll see what happens after a few days. Now, if you do this at home, I do recommend that you put this in the refrigerator, and the reaction happens pretty much immediately. So right now we are seeing the vinegar interact with the calcium carbonate shell of the egg, and the bubbles that you see is carbon dioxide gas being given off as the eggshell gets disintegrated. Okay, it's been about 3 days since I started making, they're called naked eggs. So now these eggs have got their entire shell completely dissolved right now. So the next step that I want to do is I want to demonstrate osmosis using these eggs, because essentially an egg is a cell. So what you need is 3 Ziploc bags. You'll need K-rose syrup, which will demonstrate hypertonic activity, and then this is distilled water, which will demonstrate hypotonic activity. My control group will be an egg placed in a bag with nothing in it. And then the point of all of this is to wait a few days and see how the size of the eggs change based on the environments in which we put them in. So we'll go ahead and do that. Now they're real jelly-like, kind of gross, but we're going to put 1 in each bag. And in this one here, I'm going to put the corn syrup in. Now corn syrup is loaded with sugar, and so what will happen is over the next couple of days, the fluid inside of the egg will try to achieve equilibrium with the lack of water found in K-rose syrup. So K-rose syrup is full of solutes, and what will happen is those solutes will draw the water out of that egg, causing it to shrivel in size. The opposite one is a hypotonic environment. So I'm going to put an egg inside of a bag I'm going to fill it with distilled water. Now, distilled water has no solutes in it whatsoever. It is just plain H2O. And what we should expect is the water outside will be attracted to any solutes found within the cytoplasm of the egg itself, and then it's going to go right inside and cause the egg to swell. So we're going to give this a couple of days, and then we'll see what the results are. And don't forget, all good experiments require a control group. So we're going to keep one egg by itself. I'm not going to add anything to it so it does not change the shape. So this is what an egg looks like normally. You want to put these in the refrigerator, wait about a couple of days, and then see your results. All right, after about 24 hours, what I've done is, here's our control egg. This is the egg under hypotonic conditions, which is nothing but distilled water. You might notice that it's pretty blown up compared to the other egg. And that's because this egg has taken in excess water since there's no solutes in the original distilled water. Likewise, completely full of solutes in this K-rose syrup has caused this egg to wrinkle as a result of losing water. So here's just a way of trying to demonstrate osmolarity and tonicity using eggs. Because of tonicity and osmolarity, organisms have made several adaptations that allow them to survive these conditions. Ever seen a wilted plant? We know it is due to a lack of water, but the wilting is caused by the vacuoles, which are large compartments that store water in cells, start shrinking. Plants do best in hypotonic conditions because it's difficult to damage the plasma membrane since plant cells have cell walls outside which help to support it. This allows a plant to stand more firm so they can collect more sunlight to undergo photosynthesis. Paramecium and similar freshwater pond microorganisms are challenged by living in hypotonic conditions. They must retain solutes and expulse excess water. So if you pay attention here, there's a little thing called a contractile vacuole. And then what it does is it removes excess water, preventing the paramecium cell from bursting open. And this is very important if you live in freshwater environments because you're bombarded by hypotonic conditions. Fish are really interesting. Saltwater fish, like this yellow tang, have to deal with hypertonic conditions. Their urine is highly concentrated of solutes and less water to help retain the water and void excess solutes. Bass, being freshwater, have the opposite issue. They live in hypotonic conditions and must have urine mostly made up of water and kidneys to help retain more solutes. So it's a similar condition to the paramecium I just showed you. Up next, we'll be looking at active transport and bulk transport.